Thank you.
So I think I have half an hour, right? Okay, perfect. So my name is Hoang, and I'm a genetic counselor for the Autism Research Study. One of the best parts of my job is giving genetic feedback to families, so I'm really happy today to be here to give a genetics update from our research. Now, my primary role is in autism, but a lot of what I'm going to talk about does apply to other neurodevelopmental disorders as well. So for the talk today, I really framed it around three main questions. What do we currently know? What has research taught us? And where do we go from here? And throughout, I've tried to incorporate some of the common questions that I've received from parents from the one-to-one counseling as well. So we'll first start with, what do we currently know? So the rates of autism have steadily increased since the 1970s, with a drastic jump in the past few years. And the number of children identified with autism is now estimated at 1 in 68, with a sex difference of 4 to 1 affected males to females. We know in part the increased incidence is due to an increased awareness by professionals to recognize the early signs and symptoms of autism, as well as a change in how we diagnose ASD, and that includes a broadening of the presentations. But these two factors together don't account entirely for the huge jump, so there are some unknown factors as well. What we do know is that autism and other neurodevelopmental disorders follow what we call a multifactorial inheritance pattern. They're multifactorial conditions. And what that means is that there are environmental factors as well as genetic factors, both strong and weak, that contribute to a risk. So you can imagine that each of us has a risk for developing autism, which is represented here by this empty cup. And it's not until you have enough risk factors to fill the cup that you would have a diagnosis. So each and every one of us has some of these factors, but it's only until enough risk factors overflow that cup or reach what we call a critical threshold that one would develop autism. So for the majority of my talk, I'm going to talk about the genetics, but I did want to take a minute just to talk about environmental factors. I've received questions in the past from parents like, was it something I did? Was it something my child was exposed to? Is there something I can avoid? So this year there was a study that was published in Health Promotion and Chronic Disease Prevention in Canada that was a review of all the studies conducted on environmental factors. And really they grouped the different factors into these 4 categories, the bigger balls representing the factors that have been studied more commonly. And so physiological factors have been studied the most, and they include things like parental age, pregnancy-related factors, maternal conditions, birth order and birth spacing, as well as child conditions. Chemical factors include things like toxins and pollutants, maternal exposures, medications, and vaccines. Social factors are things like stress level, usually in the parent or in the mother, immigration, and social economic status. And then finally, nutrition, which hasn't been studied a whole lot, but include things like infant feeding and vitamins. So I've thrown up a few examples here just because I think you may have heard of them in the news or in talking to other parents. Collectively, the take-home message about environmental factors are that they are difficult to study for a number of reasons, including the timing at which the exposure occurred. How specific is it? Is it just one factor, or really is it something else or a combination? And then are there confounding factors? Is really what the result that's found really due to what they're looking at, or is it something else? And overall, the number of studies on each of these kind of topics varies, and the quality of the study varies, and therefore the results tend to be inconsistent. One study may show an association, another study may show there's no association. But overall, what we do know is that the absolute risk for environmental factor is small, meaning that they don't cause autism, but they contribute to the risk. So they're a small ball in that cup and wouldn't fill the cup entirely. So I just want to take a moment to highlight just 3 that have been studied more commonly and have more consistent results. So one is the age of the father. So studies have shown that older fathers tend to have a slightly increased risk of having a child with autism compared to younger fathers. Again, absolute risks are small. Vaccines, including the MMR vaccines and the mercury component in vaccinations, have been widely studied, and the studies that have been better designed are consistent in showing that it's not associated with autism. And then finally, valproic acid, which is a medication that's used in seizure treatment, has been shown to be associated with an increased risk. And again, the risk is small. The absolute risk in this particular example is 5% chance. So now I'm going to move on to genetics, which is really the biggest focus because it plays a very strong role in contributing to autism. It accounts for 70 to 90% of the risk. To understand genetics, I have to first understand genes. So we start with the human body. So our human body are made up of cells. We have billions of cells, heart cells, brain cells, skin cells. And in nearly all of the cells is the same set of genetic information that tells that cell how to grow and function properly. That's our genetic sequence, our DNA. And you can actually see them in the cell. They're organized into these structures that we call chromosomes. And when you unwind the chromosome, you'll see that that's our genetic information, and there are sections that are called genes. Genes are important because they are the instructions for making proteins, which you can think of as workers in the body. They have a specific job. So we have workers, proteins that work in the brain, proteins that work in the heart. We have about 25,000 genes that code for proteins. We don't know what they all do, but we do know what they are. So when we're talking about genetic risk factors, what I'm referring to is a change in the genetic instructions so that the protein may not be made properly and therefore may not work properly in the body. Or it's not made at all, and then it's not working and missing completely from the body. So we can look for genetic variants using a blood test because blood is a source of blood cells. And in the blood cells, if we looked at them under the microscope, you can actually see that there are those chromosome structures that hold our genetic information. And when you organize those chromosomes by pattern and by size, you'll see that we have 46 of them, and they come in pairs. And so the 25,000 genes that I was talking about are spread across those different chromosomes. One copy of every chromosome comes from our mom, and one copy comes from our dad. And in males and females, they're pretty much the same except for the last chromosome pair. You'll see here at the bottom there is an example of a female with XX, and a male is XY. And so genetic testing can identify genetic variants, and really the technology has evolved quite drastically over the years to allow us to detect more genetic variants and different types of genetic variants. So we start off with a karyotype, which is a genetic test that's routinely used in the clinic now, and moved on to microarray technology, which is also a clinical test that's used in testing individuals with autism. And then now we have access to whole exome and whole genome sequencing, which is primarily still used in the research setting. So to understand what each of these tests pick up, if you start off with a chromosome, the chromosome pair I was talking about, and imagine that they're books. So each chromosome is a copy of a book. You should have 2 copies of every book, one that mom gave you and one that dad gave you. So just by looking at the chromosomes is what karyotyping does. So if you're looking and you can see that there is an extra book, a missing book, or in this case, a missing part of a book, you would be able to see that. So karyotyping is a genetic test that detects large structural chromosomal changes. And when we test individuals with autism, we see that about 5% of the time, these individuals will have large structural changes that can be picked up by karyotyping. Microarray technology is a little bit more detailed. It goes one level deeper. And so you don't do anything to the actual book, but you might open it up and either rip out a couple of pages or insert a couple of extra pages. So these are referred to as deletions or duplications, and collectively they're called copy number variations. So microarray, essentially what it does is it's able to pick up any deletions and duplications in our genetic information. And in autism, that accounts for about 10% of the detection. And then lastly, whole exome and whole genome sequencing, which has become available only in the last year or two, is going one step further. So not ripping out pages, not inserting pages, but actually going in and rewriting that genetic information. And it's able to detect as detailed down to as a single letter change. So in this example here, if this is the instructions for making a protein, or in this case beef stew, a single letter change could change how that stew turns out or how that protein turns out. And so we've been using this technology in research in autism, and many studies have come up with different ranges, but it's about 25% to 40% depending on who you're studying. So the next section is what has research taught us? Okay, so when we look at individuals who have autism and look for genetic variants, what we see is a different combination in each and every individual. We're all unique, and individuals with autism are unique from a genetic point of view. And when we look at the genetic differences, we see that there are different genes involved, and actually there are hundreds of autism genes. And when we plot them or see where they're located, you can see in this diagram that the genes are spread across all of the chromosome. There's no one chromosome that is an autism chromosome. So there are hundreds of genes. There's 75 to 100 that we refer to as large impact genes, meaning they're big balls in the cup. And there are hundreds of smaller impact autism risk genes, which are the smaller genetic balls in the cup. What we know as well is that genetic factors can be inherited, like in this example in child 1, where the autism threshold is reached because of a combination of inherited risk factors, both from the mother and from the father who don't have autism. And genetic risk factors can also be new in the child, what we refer to as de novo. And this is illustrated in child 4, where the majority of the autism risk is attributed to a large new change that happened. And in a recent study that we conducted, when we enrolled families who had more than one child with autism, we found that more than half of these families, the 2 children who had autism, had different genetic variants, like in this diagram here, which was a bit of a surprise because we thought if it's running in the family, they share same genes, they should share the same genetic underlying risk factors, but that's not always the case. And what we also found is that no one genetic variant or even one gene accounts for more than 1% of autism. So if you have a room filled with autism, there's not going to be the same gene involved in more than 1% of the individuals in that room. It seems to be these kind of unique hubs. So then when we looked at what the hundreds of genes actually do, what do they code for, what do the proteins do in the body, we find that they interact with each other and in these common biological pathways. And that's illustrated here. So some of the genes code for proteins that are involved in synaptic and neuronal adhesion components, which just refers to the fact that they have a role in making connections between brain cells and brain signaling and communication. Some of the genes play a role in transcriptional regulation, which is a process that controls how genes are expressed, which ones go on to make proteins. And then some of the genes code for proteins that play a role in RNA processing, which is a process for how proteins are made. So these are the three main biological pathways that seem to overlap the hundreds of genes. So one of the questions I often get from families is, how likely will you find a genetic variation if I do this research study, and when will I get my results? I've referred to this kind of as detection rate. So using whole exome and whole genome sequencing and scanning the 25,000 genes, we find 3 to 4 million variations. That's a lot of variations, and we know that not all of them contribute to health and developmental difficulties. Some of them certainly do. The majority do not. They are called normal variations or benign variations. And then there are ones where we don't quite know. They might be playing a role in contributing to health and developmental difficulties, or they might not. We just don't have enough information. So they fall into this category of uncertainty. So the time it takes us to actually run the sequencing and pick up the 3 to 4 million variants is time consuming, but the most time consuming step is categorizing the variations that we find to filter out the ones that are important. And that often takes years for us to do. And in part because of this uncertainty factor that there isn't enough information currently available. But as we do more and more sequencing, we'll hopefully have more and more data. And so in about half of our families, we do find something that's worth reporting back, and in which case a report is generated and you would meet with me for counseling, or your team who enrolled you. So another question I get asked often is, what is the chance of having a second child with autism? And so for those families where we have a genetic result, we may be able to refine that recurrence risk information for the family. And that really depends on what we find. Is the variation inherited from a parent? Is it de novo? What are the genes involved? What chromosome it's on? So using that information, we may be able to tell that family what the likelihood that the second child would have the same genetic variation. However, it's important to note here that we're not able to tell what is the chance of that child going on to have autism, because remember, there are other factors involved. And then as well, even if the child has autism, we can't tell at this current point in time where they'll fall on the spectrum. So those are some of the limitations. For families where we don't have a genetic result, or the result came back and there was nothing that we filtered out to be contributing, we have to rely on empirical data. And this is information that's generated from studying other families that have a child with autism, and they essentially count to see how many go on to have a second child with autism. So it's not specific, it's more collective or general data. And according to a recent large longitudinal prospective study, the estimate is about 15 to 20% chance of having a second child with autism when you already have one child with autism. And this risk estimate is not based on how severe that child who's affected is, or whether it's a boy or a girl. It does change, though, if the sex of the second child or the pregnancy is male, because we know males are at increased risk. And it does increase if there's more than one affected child already in the family. So in this study, it's about a 2-fold increase. Unfortunately, we don't currently have any estimates or information about recurrence risks when the individual has autism themselves. And so this is an area that ongoing research is trying to focus on to get these numbers, but we don't have it yet. And then so lastly, I want to focus on where do we go from here. So when we generate data from research, the ideal is that it'll eventually trickle down into clinical care so that families like yourselves can use it and improve the health and development of your child. And unfortunately, the system seems simple, but it's more like this, where it's a bit more complex. It has a lot of steps to go through, and oftentimes it takes a long time for information to be beneficial. And when you add genetic information into that picture, and autism, which is so complex, and as well as neurodevelopmental disorders in general, it generates a lot of data that often overloads the system and creates gaps in the process. Some of these gaps I've listed here for autism. So one of them is an unclear genotype-phenotype correlation. Another one is limited evidence, as well as uncertain biological impact, multiple factors involved, and unclear treatment benefits and risks. So all of these gaps in our understanding means that only a little bit of information ends up trickling down into clinical care. So let me take a few minutes to go through each of this in a little bit more detail. So unclear genotype-phenotype, what we mean by that is we know that there are genes which are implicated in more than one condition. So for example, a genetic variant in a gene called astrotactin 2 has been implicated in autism, but also obsessive-compulsive disorder, as well as attention deficit hyperactivity disorder. So it's a risk factor for all three of these conditions. And then this individual, the threshold is reached for autism, so this child presents with autism. In another individual, the child may present with just ADHD. And in the third child, they may present with autism and OCD. These are just some of the combinations. So it's unclear what the actual presentation is like for every genetic condition, and we need to further study that. There's also limited evidence because the variants that we're finding are rare. They've only been reported a few times in the past or not at all. So it makes it hard for us to make a call. Is it playing a role then, or is it not playing a role? So the significance is uncertain. And part of that uncertainty also lies in the uncertainty behind the biological impact. We know a variation is there, but variations don't have to be disruptive. So is the variant actually doing anything to that protein? And if it is that protein then not doing what it should in the body, and therefore the body is impacted? And there's unclear treatment benefits and risks. So let's say we find a genetic change in a gene that codes for a protein that's responsible for pumping out these molecules, which is important for making connections between neurons or brain cells, which is vital for the normal functioning of the brain. So let's say, okay, let's put in the missing component or the downstream effects. Is there a drug to do that? And if so, when do we do that? Timing needs to be considered, as well as dosage. So all of these kind of unclear benefits and risks are a potential challenge as well. And then lastly, multiple factors are involved. I already talked to you about the multiple genes that we've identified. I covered a little bit about the environmental factors, but there's also epigenetic components playing a role. And what I mean by that is that there are genes that are called epigenes. And so these genes actually control how other genes work, where then the gene that's impacted may have a lesser role in contributing to risk or an increased role in contributing to risk. So it has kind of this manager relationship where it manages and regulates how other genes are turned on and turned off. So to tackle and repair this system of translating research information into clinical care, we need a lot more research, and that's currently being done, and future initiatives are underway as well. So one of the things we want to do is look to do more case reports or case series. We want to develop better databases, utilize stem cells, run epigenetic analysis, and use animal models. So I'll go through each of those as well. Still okay. So to tackle the unclear genotype-phenotype question, what we want to do are case reports. What that means is we invite families who have a specific variant or in a specific gene and assess them to see what else do they present with. So is it just autism alone, or do they have ADHD, OCD, maybe they have seizures or other health complications, to get a better understanding of the breadth of presentations that can be part of that underlying variation. The limited evidence, we can tackle that by creating databases to pool genetic information. So we might only have one family with a variation in a specific gene, but there might be other researchers from around the world that have a family. So can we pool that and combine that knowledge? And we're currently doing that with a project called Missing, which is a partnership between Sick Kids, Autism Speaks, and Google. And the goal is to upload 10,000 whole genome sequencing data, as well as the phenotyping, the presentation data, into a database that other researchers from around the world can have access to. And as of October of this year, we have about 7,000 families, or 7,000 genomes now in that database. For the uncertain biological impact, we want to utilize stem cells, and specifically what we call iPS cells, which stands for Induced Pluripotent Stem Cells, to study how specific variations impact that cell. So what I mean by that is we're inviting families to either give us skin cell sample in the past, and currently a blood cell sample. And what we do is we take that blood cell, for example, we revert it into a stem cell, and then we induce it to become a brain cell. And the idea then is we have a brain cell with a variation that we're interested in looking at with that genetic difference to see, is that brain cell different in its structure or its function? So we're currently doing that, and we've seen things like brain cells that show more branching than it should, less branching than it should, more connection or overconnectivity and underconnectivity, and really depends on the variation or the gene that we're looking at. So we want to utilize and continue to utilize that to study what is the impact at a cellular level. I do want to point out here, because there have been questions about stem cells in terms of treatment, and we're currently not doing that for treatment purposes. So we're not taking stem cells and putting them into individuals who have autism to treat them. But for the treatment line, we are using animal models. So animals where we can program them to have a similar or the same genetic variation as in the individual, and then test potential treatment medications and drugs to see, are the symptoms in that animal improved, and are there any adverse effects? And then finally, to tackle the complexities of autism and neurodevelopmental disorders, we want to do epigenetic analysis. So this is exploring how variations in epigenes impact the expression of the other 25,000 genes to see, is there a difference there? So the hope is that once we can fill the gaps in these fill the gap in our understanding with these additional research, then eventually we'll be able to translate it into clinical care. So with my last slide, I actually went through that pretty quick, I want to tackle what the potential for clinical care would mean. So if we're able to subcategorize individuals with autism based on their genetics, then we can inform medical management and direct the care for these individuals, and also provide parents with better information about what to expect and how to care for their children. We may be also able to identify individuals that are at risk for developing autism based on their genetics, and initiate either earlier intervention or more tailored, more intensive intervention based on the profiles that we generate. And then lastly, we'll be able to guide the development of targeted medications, so facilitate pharmaceutical intervention actually based on the genetic or the biological pathways that are involved. And so with that, I'd like to thank you for staying. So you're talking about families where there is a genetic variant that's contributing to the autism, and the daughter or one of the siblings doesn't have the variation.
No, we all have it.
Yeah.
What do they do for genetic testing later if they decide to have kids?
She doesn't have the phenotype.
Study,
Yeah.
but then you are gone to live your life.
Yeah.
When you're in the future, this sibling that does have the gene
Okay.
is a female.
Yeah.
Is there genetic testing? What happens
Yes.
with parenting when they're older?
That's a great question, yes. So a lot of our participants are coming to the age of considering reproducing and family planning. So it really will depend on what the result is, how big of a ball it is in the model, the cup model. If we think it's a significant risk factor, one of the first steps that we would do for research families is clinically validated. So it's an important step to make sure what we found in the research is actually there. And also a lot of the clinical potential services that are available need that confirmation. So assuming that it is a strong risk factor for autism, one of the bigger balls, then the individual would have access to potentially prenatal testing, potentially what we call pre-implantation genetic diagnosis, and so there are additional genetic testing that can be offered in pregnancy or even before pregnancy. So with that, you would, for the clinical validation step, you should coordinate with your research group, so Sir Pond, and then from there, it's usually a referral to a clinical center, and that can be done, again, facilitated through us or a family doctor. No, so I think that's an important part in it's what we call dephenotyping. It's actually better characterizing how it presents. So one of the things that we often come across is a child with kind of the typical autism. We bring the entire family back in to assess them, including siblings and parents, and we find kind of some symptomology in parents and siblings as well. And some of them may be attributed to the having the variant or not having the variant. So I think that's a big step in this gap that I'm talking about, is doing these case series to really highlight that it is a wide range from normal to atypical. And so it's better characterizing to better inform families what to expect. I think genetic testing shouldn't be taken as just because we find it doesn't mean we need to avoid it. I think the motivation behind the research is really to understand the breadth of it. Yeah, that's a great point. I didn't mention that, but it's a moving target. So it's dependent on the amount of data that we currently have. So the idea is that in the future, we'll have more data, we'll have more families, and we'll be able to reassign that classification to either playing a role or not playing a role. And so the actual sequencing data stays the same. We're not going to repeat the sequencing. So the data is there and is continuing to stay in missing and in our internal databases, so that as we gather more information, we can do the reclassification. Oftentimes, that's done not systematically, but kind of as we find out more cases, we'll then go back and say, oh, actually, we have 5 other families that had this. Let's bring them back in, see what else needs to be done, and then have that communication, whether it be that discussion about, now we do think it's playing a role, and here's what you need to do because of it.
Okay,
Yeah.
I just want to clarify for myself, like if we haven't heard from one in X amount of time,
Yeah.
doesn't mean we won't hear from them.
Yes, so not hearing from us doesn't mean that we ran the test and there's nothing. It could mean you're in the pipeline, or it could mean we haven't pulled out anything. There's multiple reasons why you haven't. We do have things in place to kind of flag the ones that we need to bring back. So we will be in touch if there is anything, and that continues as long as the study goes on. So that's why it's important also to keep your contact information up to date with us, because it does take a very long time.
I mean, just to give you 2 examples. The first one is the consent that you guys signed for genetics is what we call open access consent. So there was language in there that you told us that we can keep your information for a very long time. You have to tell us if you want it removed. Otherwise, we have it There is no expiration date. But just to give you an example, this year we returned results to somebody 10 years after their last communication, right?
Yeah.
10 years ago we knew nothing, we know something very strongly today, so we return results 10 years later. So your data stays with us. If we have something, we call. If we haven't heard from us, we just don't know.
Mm-hmm.
Right?
At the back there first, yeah.
You have the first testing done, I think it's called
Karyotyping, yeah.
and then, or the microarray.
Mhm.
Is there a benefit to having other testing done now that the genome testing? Will it tell you anything that the others do?
Yeah, so the evolution of genetic testing has allowed us to find different genetic changes that would be missed from the previous generation of testing. There is some limitations on what the most in-depth genetic testing can pick up. So whole genome, whole exome sequencing really finds small changes. In the past, when it was first introduced, there were some limitations where it couldn't pick up those large deletions or duplications. They've kind of ironed that out, and it seems to be able to pick it up. So I would say some of the newer testing can not replace, but does overlap with some of the older generation of testing. But there are benefits for each of those testing. For example, karyotyping is able to find rearrangements in the chromosomes that some of the microarrays and whole exome sequencing can't pick up.